So I want to tell you about the story about CIR6 specific in the brain. And first of all, a little bit of introduction of what we do in the lab. In the lab, we are interested in aging. And we are used to see aging as a normal thing. But it's really interesting what is happening at the molecular level. Why do we age? And the problem with aging is that it comes with age-related disease. We get more cancer, more neurodegeneration, more heart disease, metabolic disease. So aging per se is not a problem, but all the diseases that are coming with aging. And one of the most accepted theories about aging states that as we accumulate more DNA damage, this is what is causing the organism to age. So what happens when there is DNA damage, and particularly double-strand breaks are the most dangerous type of damage because we can lose genomic material and we can also have a genomic instability by translocations. So first we have to recognize the signal that there is damage. Then, this is a little bit weird. Okay. Then we have the sensors that will activate this pathway. These are several kinases that can phosphorylate hundreds of proteins that will give us the cellular response to it. So in some cases, we activate the checkpoints and we repair the cells. We need to modify chromatin and also decide how we are going to spend the energy of the cell if there is too much damage, if we have to stop other things and use the energy just for repair. And in worst case scenario, we can decide to go to senescence or apoptosis, and this will cause degeneration of the tissue. But also a very bad scenario could be that the cells keep dividing and then we could get cancer. So DNA damage could cause all of these problems. And in the lab, we work with a family of enzymes called CIR2s that were first discovered in yeast by their deacetylating activity. They can deacetylate histones and proteins, and in this way, compact chromatin. They are linked to the metabolic status of the cell because they are NAD dependent. And in mammals, we have 7 CIR2s that share the core catalytic domain, but have different subcellular localizations. Some of them are nuclear or cytoplasmic, some in the mitochondria, and some in the nucleoli. And their deacetylase activity or ADP ribosylase activity or deacylase activity, they can also remove fatty acid groups, is different among them. And the mouse phenotype of the knockout enzymes is very different. And in the lab, we work with CIR6 that has a very profound phenotype. Mice without CIR6 in the whole body present a progeroid syndrome. They have like a premature aging phenotype. And they have this lordokyphosis, the curvature of the back, and they have loss of subcutaneous fat and colitis, and they die from hypoglycemia by 2-3 weeks of age. And at the mice overexpressing CIR6, this was done by Chaim Cohen's group, they are long-lived. They live about 10% more in the males. So what is happening at the cellular level? And at the cellular level, we see that there is a lot of genomic instability. So just to make a very long story short, during my postdoc, we investigate the role of CIR6 in DNA repair. And we saw that by 5 seconds of DNA damage, CIR6 was already recruited to the sites of damage, and it will bring another chromatin remodeler to the sites of damage. And together they will open chromatin to allow the recruitment of all these downstream factors, downstream repair signaling that allow repair, such as 53BP1, RPA, and activation of gamma H2AX. So this is the model we came up with in the postdoc. We have DNA damage. CIR6 is recruited very fast and brings SNF to H. They deacetylate H3K56 and allow the opening of chromatin and recruit the downstream signaling that allowed repair. If not, we cannot repair and we accumulate damage. So what happened at the physiological level where this is relevant? And we look at different tissue, and we saw that in the brain, for example, if we don't have CIR6, SNF2H is not recruited, and it comes with hyperacetylation of K56, but also with all these signs of DNA damage, such as phosphorylation of ATM, Caspase-3 cleave, P53 phosphorylation, and PARP cleave. But if we look at other tissue, such as muscle or liver, this didn't happen. So this is tissue specific. Not all the tissue depends on CIR6 and SNF2H for repair. And why the brain is so important and the DNA repair is so important in the brain? First of all, because it's a very metabolic tissue. It weighs about only 2% of our body weight, but uses 20% of the oxygen and 25% of the glucose. And this can generate much more metabolic damage. Also, we need to keep these neurons for a lifetime. Sometimes we need to keep them for 100 years. So this DNA damage accumulation can be very severe in the brain. And another information, another clue for the importance of DNA repair in the brain comes from inherited mutations that affect the DNA repair enzymes. You can see here where there is a little brain, it means microcephalia, neurodegeneration, ataxias. These enzymes are affecting double-strand break repair, nucleotide excision repair, single-strand break repair, and you can see there are many. So DNA repair in the brain is very, very important. And this is why we decided to do a specific SIR6 knockout in the brain. So the whole body knockout died by 2-3 weeks of age. So we developed, after Sugar Group, a brain-specific SIR6 knockout. They show that they are born smaller, but as they age, they become obese when they reach like 1 year old. And first, we decided to test these mice for their behavior, their learning capacity. And I'm showing you here one of the tests, but we did several tests. This is the contextual fear conditioning, where you put a mouse in a cage and it's not afraid of the cage. But if you give an electric signal, it will be afraid of this cage, but not in another cage. But if you couple it to a sound, it will be afraid of both things, the cage and the sign. And you can see here that wild-type mice learn and they freeze, but knockout mice don't learn at all. And these are 3-month-old mice. These are very young mice. And we did also the open field, the novel recognition object, and several other behavioral and learning tests, and they are impaired in all of them. So our mice are really not the smartest one or not learning. And when we look at the brain and what is happening at the brains, we can see that there is a lot of DNA damage signaling, DNA damage accumulation. We see hyperacetylated K56 and phosphorylation of ATM. We see gamma H2X phosphorylated, and we see the lack of recruitment of SNF2H to chromatin. And we see an increase in apoptosis in the cortex of these mice. So we decided to generate a cellular model so we could investigate the molecular causes of this phenotype. So we generated by CRISPR SHSY SIR6 knockout cells. And you can see that our cells really resemble the brains of these mice. We don't have SIR6, we cannot recruit SNF2H. We have increase in PARP cleaved, hyperacetylation of K56. And we generate also some other lines that were so impaired by the lack of SIR6 that they couldn't even divide. We could not use them because they were really impaired. So next we decided to check if these cells were really more sensitive to genomic, to DNA damage, such as irradiation, and you can see that these cells are dying more. This is with the caspase 3 detector that moves to the nuclei. But if we reintroduce SIR6, these cells are rescued, but not the catalytic mutant, the HY. So SIR6, lack of SIR6 is impairing the viability of these cells and the response to damage. And one of the markers of neurodegeneration that is one of the most common in several diseases, in 17 of the neurodegenerative diseases that we know, is hyperphosphorylation of tau. And I don't have to explain all of the roles of tau in here, but just to say that it has a roles in the, also in transport to the synapses, but there are some new roles of tau also in the nuclei. And we decided to check if our mice present tau or hyperphosphorylated tau, that this is the one that is known to be pathological. And you can see that our mice, these are again young mice, 3 to 4 months old, have hyperphosphorylated tau in the brains. You can see also in 2 different antibodies, 88 and serine 199. And we decided to check, okay, which kinase, how is this pathway activated? We have more DNA damage. Why would we have a neurodegenerative phenotype? Why would we have hyperphosphorylated tau? So we decided to look for kinases, and one of the kinases that can phosphorylate tau is GSK. And GSK is inhibited when it's phosphorylated, but becomes active when it's dephosphorylated, and you can see in these mice brains that there is less phospho-GSK. And we think that as we accumulate DNA damage, GSK become activated and it hyperphosphorylates tau. And also we can see that CIR6 can directly bind to GSK. So what happens with GSK? Can we inhibit GSK activity and rescue this phenotype in our cells? And you can see here that knockout cells treated with lithium that inhibits GSK, we have rescued the hyperphosphorylation phenotype and also the apoptotic phenotype. These cells are dying less. The other thing we wanted to make sure is CIR6 has many roles. So is this related to its role in DNA repair, or it's related to some of its other roles? And you can see here wild-type cells have very little tau. CIR6 knockout cells present a lot of tau phosphorylated, but also total tau. So it's not only the phosphorylation, but also the stability of the protein that may be affected. And as we irradiate the cells, we see that wild-type cells also have increased levels of tau. Irradiation is elevating the levels of total tau and phosphorylated tau. So we think that tau stability is increased by this phosphorylation. And to prove this, we inhibited the proteasome with MG132. And you can see that the wild-type cells have very little tau, but knockout or proteasome-inhibited cells have all the same levels of tau. So really, CIR6 or irradiation are stabilizing tau protein. And to understand really the relevance of this serine phosphorylation, we did point mutants. And this experiment we repeated many times, and also the DNA we prepared several times because it was so shocking that by a point mutation in serine 199, the one that is phosphorylated by GSK, we lost all the tau protein in the cells. And not all of the protein. If we look at the microscope, because this is with the Emerald, with GFP, you can see that there is a little bit of protein, but it's immediately degraded. So CIR6 is affecting not only the tau phosphorylation, but also tau stability in the cells. So how is this related with DNA damage? And in here again, you can see that CIR6 knockout cells have activated GSK and have more total tau. But if we inhibit ATM, that is one of the most important enzymes in this DNA repair cascade, we can see that there are a little bit more phosphorylated wild-type and knockout cells. But as we irradiate, all of the GSK becomes active, and the phosphorylation of GSK is a little bit prevented, the dephosphorylation by caffeine, but not completely. But tau, it remains still in high levels. So it's not only by ATM, but we can also prevent cell death when we rescue our cells by inhibiting ATM in a short term. In the long term, the cells will all die if we inhibit ATM and CIR6. So what we think is happening is that as we don't have CIR6, we accumulate DNA damage. This activates ATM that can activate GSK and will have hyperphosphorylated tau. But there is another alternative pathway that we don't know yet what it is, that is causing also without the ATM, the stabilization of tau in the cells and in the brain. Last, we wanted to see if this is really physiologically relevant in Alzheimer's disease and other diseases that are related with this tauopathy. And you can see in AD patients that we have a really lost CIR6 expression. This is at a protein level, and we can see also at the mRNA level from data sets that were already published in the hippocampus and temporal cortex. And this decrease is correlated with the Bragg stage of the disease. As we increase in the Bragg stage, we have less CIR6. So what we think is happening is that as we have CIR6, we are young and we express these repair proteins, we prevent double-strand breaks, GSK is inactive, and tau is degraded. But as we accumulate DNA damage, this signaling occurs all the time, and we increase tau phosphorylation and tau stability, and this is happening over time. We are accumulating it over time. So if we are young or a wild-type mice for CIR6, we repair our DNA and we are okay, our brain is functioning, and we have not cognitive decline. But as we get old, CIR6 levels are reduced, or particularly in Alzheimer's disease. And then we activate this constant DNA damage signaling that will lead to cell death and to the development of these neurodegenerative markers, and we'll get a cognitive decline. And I want to thank my group. The first part I did in Raul's lab. All the behavioral analysis was done in Amar Sahai lab by Antoine Besnard. And in my group, Shai Kaluski, a master's student, and Miguel Portillo, a PhD student, did all the molecular work that I show you for the last part. And thank you very much for listening.